Hello everyone. Today I will be discussing about transgenic plants. So, why to create transgenic plants? When there is no naturally genetic variation for the target trait. For example, glyphosate, herbicide resistant in soybean, corn or vitamin A in rice, blue roses, etc. So, what is transgenic plants? Transgenic plants, also known as genetically modified plants or GMO, that is genetically modified organism, is a plant that has had its genetic material altered through genetic engineering techniques. This modification involves the introduction of specific genes or DNA sequences from another species into plant's genome, resulting in the expression of new traits or characteristics that the plant would not naturally possess. These introduced genes can come from unrelated organism, including other plants, animals, bacteria or even synthetic sources. Now, the techniques of genetic modification. There are two techniques, traditional technique and advanced technique. In traditional breeding, in natural variation, traditional breeding relies on natural genetic diversity within a plant species or closely related species. It involves crossing plants with desired traits to create offspring with a combination of traits. It is a time consuming. It can be a time consuming process because it relies on natural genetic recombination through generation of selective breeding, limited crossability. The success of traditional breeding depends on the ability to crossbreed closely related species. Incompatible species may not be crossbred easily. Unpredictable outcomes. The outcome of traditional breeding can be unpredictable due to the random assortment of genes during meiosis. Its safety and acceptance. If we see the safety acceptance, traditional breeding is generally considered safe and widely accepted as it mimics natural processes. Limited to existing traits, traditional breeding can only work with traits present within the existing genetic diversity of the species. It cannot introduce entirely new traits from unrelated organisms. You can see the whole process in this pictorial representation. Then genetic modification. Precise trait introduction. Genetic modification allows for the precise introduction of specific genes or traits into plant's genome from unrelated organisms. Rapid development. GM can produce desired traits more rapidly than traditional breeding methods. It enables the transfer of genes from unrelated organism, expanding the range of traits that can be introduced. It is having the predictable outcomes also. Genetic modification allows for more predictable outcomes because the specific gene or trait is known. It is having the potential risk also. GM has raised concern regarding potential environmental risk, unintended consequences and long term effects. If we see the regulation and public perception, genetically modified crops are subject to strict regulation and vary in public acceptance depending on the region. In some state it is accepting and in some state it is not accepting. So, labeling and transparency are key issues. Genetically modified plants can be engineered to produce pharmaceuticals, biofuels and other industrial products. In this way, we can reproduce genetically modified plants. Now, next tool is genetically engineering tools, that is the advanced technique through restriction enzyme. Restriction enzyme, also known as restriction endonucleases, are proteins that cut DNA at specific recognition sequences. They are used to generate DNA fragments with precise ends, which can be combined with other DNA sequences. In this way, we cut the restriction enzyme, and with this tool we can modify, we can prepare the genetically engineered plant. Next is plasmid vector. Plasmids are small circular pieces of DNA that can be used to carry and replicate foreign genes. Scientists can insert the desired gene into a plasmid vector, creating a recombinant plasmid. Next technique is through polymerase chain reaction or PCR. PCR is a laboratory technique used to amplify specific DNA sequences. It allows scientists to create many copies of a particular gene for further analysis or cloning. So, all three processes you can see in this pictorial representation. Next is agrobacterium mediate transformation. Agrobacterium tumefaciens is a bacterium that naturally transfers a piece of its DNA, that is TI plasmid, into plant cells, causing the formation of tumors. Scientists have harnessed this ability to introduce desired gene into plant genome. Modified agrobacterium strains are used to transfer recombinant plasmid into plant cells. You can see in this pictorial representation the procedure for plant transformation, how we can develop it. Next technique is the biolistics or particle bombardment. This technique involves coating microscopic gold or tungsten particles with DNA and shooting them into plant cells using a gene gun. The DNA is then integrated into the plant's genome. You can see the instrument here, how we are bombarding this. Next technique is the electroporation. Electroporation uses electric pulses to create temporary pores in plant cell membrane, allowing the introduction to foreign DNA. In this picture, you can see how they are doing. Next is Sanger sequencing, that is in DNA sequencing, developed by Frederick Sanger in 1977. It was the first widely used method for DNA sequencing that involves DNA replication with modified nucleotides that terminate the growing DNA strand when incorporated. Fragments of varying length are generated and their sizes are determined by gel electrophoresis to deduce the sequence. That is the Sanger method. This method is very common method. Now, next generation sequencing, that is NGS, we can say it NGS or next generation sequencing, also known as high throughput sequencing. NGS methods allow for the simultaneous sequencing of millions of DNA fragments. Common NGS technology include Illumina, Ion, Torrent and PacBio. NGS relies on parallel processing where DNA fragments are sequenced in a massively parallel manner and then assembled to determine the complete sequence. This is the workflow of next generation sequencing. Now, third generation sequencing. These technologies like Pacific Biosciences and Oxford Nanopore can sequence long DNA molecules in real time without the need for DNA amplification. They work by measuring changes in electrical current as DNA strands pass through nanopore or by recording fluorescent signals as DNA bases are incorporated during replication. The flow chart of this sequence clearly for third generation, first generation, second generation and third generation you can see in this pictorial representation. Now, next is gene isolation. The steps of gene isolation, how we do it. So, for DNA extraction, this source can be genomic DNA extracted from cells or tissues, plasmid DNA or synthetic DNA fragments. Cells or tissues are typically lysed or broken or release the DNA. Protein and other cellular components are removed through various methods, leaving behind purified DNA. Then we will identify the target genes. So, how to identify the target genes? Before isolation, the target gene must be identified based on its known sequence or by using molecular biology technique like PCR with gene specific primers. For enzymatic digestion, enzyme known as restriction endonucleases, that is restriction enzyme, are used to cleave the DNA at specific recognition sequences. The choice of restriction enzyme depends on the location and frequency of their recognition site in the target gene. Next is gel electrophoresis. The resulting DNA fragments are separated by size using gel electrophoresis. A gel is prepared and the DNA samples are loaded into wells at one end. When an electric field is applied, DNA fragments migrate through the gel with smaller fragment moving faster than larger one. After electrophoresis, the gel is stained with a dye that binds to DNA and is visualized under UV light. For gel extraction, the DNA fragment containing the target gene is cut out of the gel based on its size. The DNA is then eluted from the gel slice using specialized technique or commercial kit. For purification, the eluted DNA is further purified to remove any remaining impurities or contaminants. This can be done using various methods including ethanol precipitation or commercial purification kits. Amplification is optional if the amount of isolated DNA is insufficient for downstream applications. The gene can be amplified using techniques like PCR. This is the whole process, you can see clearly here. Then plasmic vectors. Plasmic vectors are small circular DNA molecules commonly used in molecular biology and genetic engineering to carry and replicate foreign DNA fragments, such as genes of interest within host organism like bacteria. Plasmic vectors serve as important tools for cloning, gene expression and various biotechnology applications. Here are key features and components of plasmic vectors. If we see the structure of the plasmid are small double stranded DNA molecules that exist separately from chromosomal DNA of the host organism. They typically range in size from a few thousand to several 100,000 base pair. If we see the origin of replication, plasmid vectors contain an origin of replication that allows them to replicate independently of the host organism chromosome. This ensures that as the host cell divides, the plasmid and any inserted DNA are also copied and passed on to daughter cells. For selectable markers, plasmid often carry selectable markers such as antibiotic resistant genes or reporter genes. These markers allow researchers to identify cells that have successfully taken up and are maintaining the plasmid. For multiple cloning site or polylinker, plasmid vector include a region called the multiple cloning site or polylinker, which contains multiple unique restriction enzyme recognition site. Researcher can easily insert their DNA fragment into these sites for cloning. If we see the promoters and regulatory elements, some plasmid vector come with promoters and regulatory elements that control the expression of genes within the plasmid. These elements can be modified to control when and how the gene of interest is expressed. If we see the reporter gene, in some application, plasmid vectors contain reporter genes such as beta galactosidase or green fluorescent protein, that is GFP. These genes allows researcher to visually or quantitatively access gene expression or the success of cloning. This is the plasmid map. You can see here the plasmid map. In transformation methods, the heat shock transformation typically used for bacterial cells such as E. coli. Cells are briefly exposed to a high temperature heat shock followed by a rapid cooling step. These thermal stress disrupts the cell membrane, allowing plasmid DNA to enter. In electroporation, suitable for a wide range of cells including bacteria, yeast, plant cells and mammalian cells. Cells are subjected to a brief electric pulse that creates temporary pores in the cell membrane. Plasmid DNA is then introduced into the cells through these pores. In chemical transformation, this is commonly used for bacterial cells. Calcium chloride or other chemical agent are used to make the cell membrane more permeable to DNA. Cells are treated with a calcium chloride solution containing plasmid DNA. Next is microinjection, mainly used for eukaryotic cells including mammalian cells. A glass micropipette is used to physically inject plasmid DNA into the cell's cytoplasm or nucleus. In liposome mediated transformation, that is applicable to a variety of eukaryotic cells, plasmid DNA is encapsulated within lipid vesicles, that is liposome, that fuse with the cell membrane, delivering the DNA into the cell. In biolistic or particle bombardment, suitable for the wide range of plant cells and some animal cell. Gold or tungsten particles coated with DNA are propelled into the target cells using a gene gun or particle bombardment device. Agrobacterium mediated transformation primarily used for plant cells. Agrobacterium tumefaciensis, a naturally occurring soil bacterium, is genetically modified to carry plasmid DNA. The bacterium infects plant cells, delivering the DNA into the plant genome. This is the whole method you can see in this pictorial representation. The next is selectable markers, antibiotic resistant genes. Antibiotic resistant genes are widely used in bacterial and eukaryotic cell transformation. Cells have taken up the plasmid containing the antibiotic resistance gene can grow in the presence of a specific antibiotic while not transformed cells die. Common antibiotic resistant genes include ampicillin, camecin, etc. Herbicide resistance genes are used in plant transformation. Transgenic plants carrying these genes can survive and grow in the presence of herbicide that would otherwise kill not transformed plants. Glyphosate resistance gene is an example. In fluorescent protein, gene encoding in fluorescent proteins like gene fluorescent protein, that is GFP, red fluorescent protein RFP, or blue fluorescent protein BFP can be used as selectable markers. Cells expressing this protein emit visible fluorescence under specific wavelengths, allowing easy identification. Then luminescent protein, genes encoding bioluminescent protein such as luciferase can be used as a marker. Cells expressing luciferase produce light when a specific substrate is added, enabling detection. In nutritional auxotrophy complementation, in microbial genetics, auxotrophic mutants, that organism that cannot synthesize a particular nutrient, can be complemented with a plasmid containing the missing gene. The complementation allows growth on minimal media lacking the specific nutrient. Now, resistant to toxic compounds, some markers confer resistance to toxic compound such as heavy metal toxins. For example, the cadmium resistant gene can be used to select for cells or organism resistant to cadmium. Temperature sensitive mutants have genes that function only at certain temperatures. Selecting for growth at permissive temperature or not permissive temperature can be used as selection method. Now, gene expression regulation. In gene regulation expression, some factors are there. So, transcription factors are protein that bound to specific DNA sequences, that is cis regulatory element in the promoter regions of the gene. They can act as activator or repressors, enhancing or inhibiting the transcription of the associated gene. Transcription factors can respond to various cellular signals including environmental cues and hormones. Promoter regions are DNA sequence located upstream of the gene's coding region. They contain specific elements such as the TA box or enhancer sequence that interact with transcription factors to initiate or modulate gene transcription. Now, RNA polymerase. RNA polymerase is an enzyme responsible for catalyzing the synthesis of RNA from a DNA template during transcription. Different types of RNA polymerase, for example, RNA polymerase 1, RNA polymerase 2, RNA polymerase 3 are responsible for transcribing various classes of genes. Now, epigenetic modification. Epigenetic modifications such as DNA methylation and histone modification. For example, acetylation, methylation can influence gene expression by altering the accessibility of DNA to transcription machinery. These modifications can be heritable and can play a role in long term gene regulation. Now, non-coding RNA. Non-coding RNA, for example, microRNAs, long non-coding RNAs can regulate gene expression post-transcriptionally by binding to mRNA molecules and either promoting their degradation or inhibiting translation. Now, post transcriptional regulation. Splicing of pre-mRNA can generate multiple mRNA isoforms influencing protein diversity. RNA processing, transport and stability also play roles in regulating gene expression. Now, chromatin remodeling. Chromatin remodeling complexes can change the structure of chromatin, making specific DNA regions more or less accessible to transcription factor and RNA polymerase. Hormones and signaling pathways. Hormones and cell signaling pathways can activate or inhibit specific genes in response to external cues or external signals. Now, environmental factors such as temperature, nutrient and stress can influence gene expression. Then cell cycle regulation. Gene expression is often tightly regulated during the cell cycle to ensure proper progression through cell division. In developmental regulation, during development, genes are regulated in a spatiotemporal manner to determine cell fate and tissue specialization. So, the application of transgenic plants are, for example, they improve crop yield, they pest resistance, they are disease resistance, they are herbicide tolerance, they are nutritional enhancement, environmental benefit we have and pharmaceutical production. These are the application of transgenic plants. So, the examples of transgenic crops are the BT cotton, golden rice, Roundup ready soybean, papaya ring spot, virus resistant papaya. So, these are the few examples of transgenic crops. So, some challenges and concerns are also there. For example, environmental concerns are there, ethical concerns are there, regulatory oversight is also there, labeling and consumer awareness is there and intellectual property issues are also there. So, these are the challenges and concerns of transgenic plants. If we see the safety and regulation of the transgenic plants, safety assessment of transgenic plants, the government regulatory agencies are there, international regulations are also there and labeling and traceability is there. Now, what are the benefits of transgenic plant? So, it increased crop productivity, it reduced pesticide use, enhanced nutritional content and sustainable agriculture. If we see the case study of BT brinjal in India, so many controversies are there. 2009, the Indian government approved the commercial cultivation of BT brinjal, making it the first GM food crop in India. The approval was based on data from field trials that indicated reduced damage by the brinjal fruit and shoot borer, a major pest of brinjal crop. Protest and opposition as the decision to allow the BT brinjal cultivation sparked widespread protest and opposition from various groups including environmental activists, organic farming advocates and some scientists. Concerns were raised about the environmental impact, potential health risk and the impact on traditional and organic farming practices. Scientific controversies are also there. Scientists were divided on the safety and efficacy of BT brinjal. Some argued that the crop could reduce pesticide use and benefit farmers, while others raised concern about potential that unintended consequences and the need for more through safety assessment. So, lack of transparency and regulatory oversight is also there. Critics pointed to issue the transparency and regulatory oversight, questioning the thoughtness of safety assessment and the influence of multinational biotechnology companies. So, the outcome of the lecture, if we see the moratorium of commercial cultivation in responses, controversy and opposition, the Indian government imposed a moratorium on the commercial cultivation of BT brinjal in February 2010. The moratorium was put in the place, allowed to further scientific evaluation and public consultation. Expert committee and public heritage. The government established a committee of experts to review the safety and environmental concern associated with BT brinjal. A series of public consultations were also held to gather input from various stakeholders. Debate over BT brinjal continued to be highly polarized, with proponent emphasized potential benefit in terms of reduced pesticide use and increased yield, while opponents raised concern about health and environmental risk. So, research is going on BT brinjal and its potential impact, both positive and negative, continued in India. So, this is about transgenic plants, their application, their techniques, different techniques and the benefit, and I have given one case example of BT brinjal also. So, thank you everyone.